Hey guys, this is actually round two. I did record this yesterday and somehow the file got corrupted, so I'm hoping for that not to happen again, as you need this in about an hour from the time I'm recording it. Let's jump right in. So, an animal, we just finished up plants, and general characteristics of plants have a description: photosynthetic, cell walls that have cellulose, have root systems, in most cases, seeds, flowers. We're talking about a plant. When you say animal, multicellular. So never do we classify an animal as single celled. Heterotrophic, they consume their calories from their environment. And eukaryotic cells. I think that goes without saying. This is important though. Ingestion is the means of nutrient acquisition. So even though they're heterotrophic, meaning they have to get their food from the environment, they don't manufacture it, fungi are also heterotrophic, but the distinguishment comes from the digestion is outside of the structure, then absorption is inside, whereas ingestion in animals is the mode of nutrient acquisition. Animals don't have cell walls. That's something that's found in plants, also found in some fungi. Most are diploid. In general, they're diploid, go through a sexual stage for reproduction. So eggs and sperm, you know that, only haploid cells. So there's no alternation of generations here like we had in plants where there's haploid adults that make haploid spores. This is it. This is what you know. The term blastula, you've probably seen in human reproduction. I can come over here and highlight it for you. And this is that hollow ball that's going to become our basic body plan. Prior to this, you've seen a term before. It's called a morula, Morula. That's the solid ball. So one of the first changes that really makes us a mammalian is when this ball opens up in the center and creates an internal cavity. And then the second hallmark characteristic in animals is that when this folds in during development, that's called gastrulation, which is when the blastula folds inwards, beginning to create an entry point. So here in this organism, which is actually a developing starfish, we see this hole opening up towards the inside. So now nutrients can flow into this individual and absorption can happen on the inside. That's the ingestion piece. So on the inside of this is called the endoderm. That's the lining. And then the ectoderm is along the outside. And anything that happens for the inside portion of the endoderm, so where our muscles and organs would be, that's referred to as the mesoderm. Our ancestry is very similar to how we talked about with plants. So as we say, plants arrived around 3.9 billion years ago. That's not plants, I'm sorry, that's prokaryotes, so life. But the evidence of life is 3.5 billion years ago. So that 400 million year gap that exists between animals, excuse me, between prokaryotes that are photosynthetic at 3.5 billion years ago and prokaryotes that are suspected to have been here 400 million years prior to that is because the photosynthetic prokaryotes were advanced to a point where they could not have been the first form of life. So we make an interpolation backwards to say 3.9 billion years ago. Well, how do we apply that to animals? There's no good fossil evidence for the arrival of animals. So the oldest fossils are at 575. But same as photosynthesis, these animals are too complex to have been the first animals. So they likely evolved from something prior that didn't leave good fossil evidence. You know, they were softer or died at a time that wasn't good for fossilization. From 575 to 542, or 33 million years, there's a dramatic increase during that Cambrian explosion. Okay? And what really was the cause of that? Over 15 million years, new body coverings, which are those new adaptations, predators had to change their game. The ones that didn't died. But feeding, ability to move, and protection are major features of predator-prey relationships. So that was occurring. Sexual reproduction was doing its thing of advancing the species, but then the global oxygen levels were at a much greater high than they've ever been before, and This allowed for the metabolism to produce more oxygen, process more calories. They became more active and more mobile because the environment was suitable. So it was like this renaissance period of mammalian adaptation. They were just really taking off. Okay. As we're talking about them, most animals are going to be invertebrates. So when I say invertebrate, I'm hoping you're thinking initially of insects. That's appropriate because insects are the most abundant of all the animals. We are vertebrates, as you know, but that makes us only one of 35 animal phyla. So phylum Chordata, that's us. 96% of them are vertebrates, right? And includes, so 4% are not vertebrates. We'll discuss that when we get there. But there's 35 animal phyla and only one is vertebrate. So that should tell you that the diversity is pretty striking. Hypothesis, not fact, hypothesis. Cells living together in groups somehow found a way to continue to live together, and in sharing some DNA, some lateral gene transfer, we end up with a reproducible colony of cells, unspecialized, of course, but just living its life. And then cell specialization had to emerge. Again, evidence of this is very, very limited because there is no good fossilized remains of these small creatures. But that had to happen. Generic cells, special cells. Now, we talked about 4 types of tissues in here. That's just one example of the specialty of cells: epithelial, connective, muscle, nervous. Even in muscle, there's 3 types. And then we can get down even further than that. So specialization was important. It was really important because prior to specialization, sexual reproduction was an issue. They had to have cells coming from sex cells. Sex cells had to come from cells that made sex cells. Gastrulation, as I've already stated, was a big piece of this puzzle. And then the reason for that is because now there's an internal gastrovascular cavity, and that can really just process calories to a better degree. All right. Animal body plan. This is the basic animal body plan. I'm drawing a diamond because I hope that it doesn't cause us any problem. Yep, good. So that's the body. There's some legs. There's a head. That's the basic body plan you think of it as an animal. Prior to that plan emerging, there was something a little bit less complex, and we're going to compare radial symmetry to bilateral symmetry here, and it's these relationships that tell us who's more or less related. So radial symmetry sounds, if you're hearing the word radius, that's good. This creature here could be a sponge. This is radial symmetry, where it travels in a radius. So what you end up with is the ability to cut in any direction and create equal portions. But it does not have symmetry top to bottom. So top to bottom, it is asymmetrical. It's radially symmetrical. So moving around its radius, it is symmetrical. In bilateral symmetry, we draw a line down the middle of the creature, equal right and lefts, and dissimilar front and rear. So the anterior end moves towards the head, the posterior end moves towards the tail. Now we're upright, which makes us absolutely different than most creatures on the planet, in that our anterior is superior or above, not forward. But if we were on 4 limbs, our anterior would still be our head. Let's see what I didn't cover here. Okay, yeah. So something I have to discuss with you here is over here. Typically sedentary, or they passively drift. So what this means is this creature here generally is going to live in water, and water moves in all directions. So the fact that it's radially symmetrical means it faces its environment regardless of what direction its environment is coming from. So water moving over the top means that nutrients can fall in. Now, if this was a drifter and was tumbling through its environment, it's the same scenario. It has equal opportunity to get nutrients no matter which direction it's drifting. Bilateral symmetry, in general, means that the organism moves towards its anterior aspect, moves towards its head, because that's where the sense organs are. Everything moves towards that, okay? Environment first. I know you're thinking about the crawfish moving backwards, and yes, it does, but it can move forwards as well. The escape mechanism is moving backwards. So anterior is the head, posterior is the tail. Dorsal then becomes the back, and ventral becomes the belly. It's the bottom in earth-facing creatures. Okay. True tissues, major advancement. Tissue, if you remember by definition, is a group of similar cells working towards a similar function. Separated by membranes, you're starting to see how there's an architecture to these animals. If you look at a building, think of just a warehouse, just a general warehouse, 4 walls and a roof and a floor. Nothing in it. That's not real special. But then start thinking about different warehouses, food processing plant, building cars, building ventilators. There's going to be special aspects of that warehouse and special compartments that do special jobs. There's the office, there's the engineering, there's the floor, there's the manufacturing plant, there's the assembly plant. So it's broken down. So animals have that on the inside of us if you want to take that mold and apply it upwards to us from the warehouses. So the earliest animals don't have that complexity. Sponges lack the true tissues, which means that they're very, very basic. That gastrulation following them gives us the specialized structures. So body cavities are another issue we look at in terms of specialization, and the cavities become more predominant in the creatures that we're going to call more advanced. Now these guys are still living, they're still effective at doing so, they're just not as advanced by these standards, okay? So what we have to highlight is the presence or the absence of something referred to as a coelom, which is here. That's the absence. This is the first time we see a cavity, but that's called a pseudocoelom because it's not attached. This coelom is detached, and there's an attachment of the mesoderm, as you can see there. And that's important because that means our organs are suspended. So protostomes, this is a term, I know it's a funky term, but we're going to get into it later as we look at this. We see protostome here. If you follow these lines, I think you're going to see that for the most part, these are things you consider to be less advanced, but please don't, because in arthropods, we've got colony warfare, caring for young, sulfuric acid secretions, stinging, pinching, flight. These guys are awesome. Without them, we don't make it. These are where the bees are, all that. So don't think that this means simple. Just thinks it means, it's different. In here are some of the less complex creatures, okay? Up here, deuterostomes, we're going to define why they're called deuterostomes later, but I just want you to see kind of where we're going with this before we get there, okay? So protostomes, the primary gastrulation, mouth forms first, and in deuterostomes, the anus forms first, and the mouth forms later. So it's important to remember as we get into this one here, okay? So I'm going to kill it there. We're going to get into the dichotomy of all of what's happening here, and just as you reflect on this today, no true tissues in the sponges. That's what separates them all from this entire group of animals. So sponges were one of the earliest forms of our animal ancestors, and we are related to them because they're animals. They just don't have true tissues. They develop differently, and as a result of not having true tissue, the cell specialization is very minimal as well. Okay. All right, squashing it there.